We'll give folks a moment to filter in here. And while everyone's coming in, I'll get us started with a couple of announcements. Hello, everyone. Good morning, good afternoon, good evening, wherever you might be. Welcome to the 8th week of the fall 2021 iteration of the Long-Term Animal Research Seminar Series. Thanks for being here. This week, we hear from André Ferreira and Dr. David Fisher, and we'll hear from André first. And before I introduce him, I'll make a couple of announcements. If you're participating via Zoom, you should see a Q&A tab on the top or bottom of your screen. If you open that tab, you'll be able to type any questions that you might have, as well as see and upvote other people's questions. At the end of the talk, we'll go through those questions, starting with the ones with the most votes. And second, as always, we'll be posting these talks to YouTube for your shortly after they conclude for your viewing and reviewing pleasure. So if you know other folks who aren't able to attend live who will enjoy these talks, please share it with them so they can watch them as well. All right, so our first speaker today is André Ferreira, a PhD student at the University of Montpellier. André previously completed his bachelor's degree at the University of Coimbra in Portugal, where he studied juncos, and then his master's degree at the University of Porto, where he began to study sociable weavers, a study system that he's continued to work with in his PhD, with a focus on helping behavior. So André has just submitted his PhD thesis, and he'll be starting a postdoc at the University of Zurich in January. But at this stage, he already has 11 publications in print or in press, so a prodigious publication record. André also, he does a great job, I think, of combining existing tools to make discoveries about social behavior, but also he's made substantial innovation by developing new methods for recording behavior. And it's one of those methods that we're going to hear a bit about today when we'll hear about the use of deep learning for individual recognition. And in my postdoc, I feel like I've just started to really be exposed to the power of automation and machine learning for collecting and interpreting behavioral data. So I'm really excited to welcome André today and hear more about his work and the future of the field. So I'll turn things over to him now.
Thank you, Matthew, for this very nice introduction. And thank you for the opportunity to be here and to present my work. So today I'm going to talk about a bit of the work that I did during my PhD on the use of deep learning for individual identification, as well as the importance of long-term projects to develop these automated systems. So individual identification is a key method in long-term projects and animal research, as it allows researchers to collect data on their study animals and study species across many contexts and across their different life stages. Usually researchers either rely on natural marks that individuals develop in their fur or in their skin, such as unique color patterns or scars, or in cases where animals are indistinguishable to the human eye, researchers rely on artificial tags, such as coloring in birds or skin and fur painting in many mammals and reptiles. These two approaches usually suffer from two major limitations. First, artificial tags raise obvious ethical concerns, as animals have to be captured and handled in order to fit these tags, which induces them with additional stress. Furthermore, these tags can also be the source of injuries, or it can make animals more visible to predators or decrease their ability to flee from them, decreasing their survival probability. Natural marks don't suffer from these limitations, but usually both natural marks and artificial tags are associated with time-consuming tasks, as researchers either rely on direct observations of the animals to collect behavior on them, or they record their study systems with video cameras for later manual and time-consuming video analysis. So how can we automatically identify individuals, possibly without even marking them, in order to make research more efficient? One possible solution for this is to use artificial intelligence and computer vision in order to make computers do the job for us of automatically identifying individuals and collect behavior on them. As a brief introduction, artificial intelligence is the theory and development of computer systems that are able to perform tasks that are usually normally associated with human cognition. And when we speak about artificial intelligence, we usually refer to machine learning, in which computers can learn from large amount of data to perform specific tasks without being specifically programmed to do them. And in recent years, a subset of machine learning, deep learning, has received much attention because contrary to classical machine learning algorithms, it does not require human intervention to point out the potential features that are important to perform a specific classification task, such as individual recognition. Deep learning is more easily understandable with an example. Imagine that you have two cats that you love so much that you have collected thousands of pictures from your two cats, and you actually organize them in different folders in your computer with the two different labels, Sushi and Percy, for example. If you then pass this data to a convolutional neural network, you can train this model to individually identify these two cats without even telling them which is the features of the cats, such as the color, the size, or whatever, that is important to distinguish them. And if you are successful, you can then show new pictures of the cat to this deep learning model, and it will be able to tell you which of the two cats you are looking at. This type of technique has been very successful in the individual identification of humans. And the reason for that is because humans in the past 3 decades have spent much of their time uploading voluntarily their own pictures to servers like Facebook and Google that are then used to train deep learning models for individual recognition. Unfortunately, there's no such thing as large-scale training datasets for non-human animals. And this is the main bottleneck preventing the widespread application of deep learning models for individual recognition of non-human animals. This is where long-term projects play a crucial role. In long-term projects, researchers mark and monitor individuals over their lifetime, collecting a huge amount of data on their study animals. This can lead to large-scale datasets that we can then later use for training deep learning models for individual identification. And we can then obtain automated methods or algorithms that are able to automatically identify individuals from pictures and videos. This in turn can benefit long-term projects, as we can automate our data collection of our automated individual data collection, and even potentially start collecting data without even capturing and handling individuals in order to mark them. In this presentation, I will talk about a few examples where this has already been applied. I will start by focusing first on the importance of marking and monitoring individuals over their lifetime. During my PhD, I have worked on 2 long-term projects, one on the sociable weaver and another on the great teeth. The sociable weaver project has started by Dr. Rita Covas and Claire Dautrelin, and has been continuously monitoring individuals since 2008 in Kimberley, South Africa. The Great Teeth project has started by Dr. Lucy Alplin and Professor Damien Farin in Constance, Germany, and has been continuously monitoring individuals since 2016. What these 2 projects have in common is that they both fit pit tags in their birds for different purposes. These pit tags are microchips that carry a unique code that is able to identify individuals whenever they land on a feeder that is fitted with an RFID antenna. This allowed me to develop an RFID camera trap that is able to automatically take pictures of birds whenever they land on the feeders to feed, and it automatically labels the pictures with the identity of the individual. This allowed me to collect a large-scale dataset of labeled pictures that were then used to train deep learning models for individual identification. I then used these models to predict the identity of individuals in different contexts, showing that these models can be generalized to multiple different contexts and potentially be used in experiments or in continuous monitoring. I will not speak in detail about this work because it has been already published in Methods in Ecology and Evolution, and you can check it out if you want to check how I trained the models and how I collected the data. What I want to mention now is also the importance of long-term projects in being authentic data libraries. A good example of this is the work of Scofield and Nagrani in the chimpanzees that use a 20-year data collection of video recordings of chimpanzees to train deep learning models for individual identification of 23 individuals. This has achieved an impressive accuracy of over 90%. Similarly, also in the sociable weaver, I have a similar approach in which I use the historic data of the project to build a deep learning model for individual identification based on the core rings. In this project, one of the central data is the nesting provisioning rates of the helpers, as we are interested in studying the cooperative breeding behavior of this species. And usually, videos from one breeding season takes between 1 and 2 years to be manually analyzed. With the help of Liliana Silva and with the 6 years of nesting provisioning data, we were able to create a data set of annotated frames from videos that were annotated with the birds going in and out, as well as their color combo combination. And by using a combination of deep learning models, and we were able to create an algorithm that automatically identifies the individuals going in and out of the nest, as well as their color combination. Although this is still a work in progress, it already made data processing twice as fast when compared with manual analysis. What these two works show is how, even when we don't have a specific data collection platform like the RFID cameras that I showed, we can still use the data that is stored on these long-term projects to train deep learning models for individual identification or to solve any other problem that we might face. I now want to focus on the current challenge and the future prospects of using deep learning for individual identification. These are the problems that I would be working on as a postdoc at the University of Zurich together with Damien Ferreira. First, I want to talk to you about age-invariant individual identification. As you know, animals change their appearance over time. Humans are no exception, and birds, for example, are also no exception. For example, on the left, you can see a picture of a juvenile sociable weaver that changes almost completely after 6 months to the adult stage. But even adults can slightly change their appearance over time, and this potentially can decrease the accuracy of the deep learning models for individual identification. To better illustrate this, I have been collecting data since 2018 on the sociable weaver, and I used data from 30 individuals from 2018 to train a deep learning model for individual identification. I then used that model to predict the identity of the same individuals in 2019 and 2020. And as you can see in this plot, the accuracy of the model decreased sharply. This is because individuals slightly change their appearance over time, and the model is no longer able to identify them correctly. One way to solve this is to look at what has been done in humans so far. And this problem has been largely solved by using data sets that contain pictures of the same person across different life stages. And this allows researchers to train models that are able to learn conservative features of the person that make it possible to identify them across their different life stages. Another possible solution is to try to understand what are the body parts of the animals that are more conservative over time. This is why I'm currently now collecting data of the birds, not only from their backs, but also from their sides, face, and so on. Second problem that I would like to mention is the fact that most research works on an open world context. So far, what I've been doing is to train models in a closed set identification problem in which all the individuals that are in the training data sets are also the ones for which the model will be used on. But most of the research is conducted, especially in the wild, is conducted in an open set identification context in which new individuals can leave or join the population at any time. The difference in training these models is that instead of training a model to fit the identity of the bird or a picture of the bird to a known identity, the model has to compare multiple pictures of multiple birds and understand whether or not they are from the same individuals. And if they are not, it can conclude that we are in the face of a new animal that's just joined the population. This approach seemed promising and has already started to be implemented on some animals, mostly mammals, with some success. The last problem that I want to mention to you is the cross-species identification problem. In some long-term projects or in some research programs, we might have access to data, or we might have the platforms that allow us to collect the data to train these models. But it might not be true for long-term projects or for research programs in which the animals are harder to observe or for which there are fewer individuals. In this case, we might not have enough pictures or videos of the individuals to train a deep learning model for individual identification. A possible solution for this problem is to use a machine learning technique called transfer learning, in which a model that has already been trained on a different species can be used as a starting point to train a model for individual identification of another species. As an illustrative purpose, I used 50 pictures on 10 from 10 great teats to train a deep learning model for individual identification of great teats. And the model is unable to reach an accuracy that passes 40%. But when starting the training process with a model that has been trained on 30 social weavers and 900 pictures per bird, the model is able to suppress 75% accuracy. This shows how promising transfer learning can be to solve the problem with sufficient data in some study models. In sum, I just want to highlight the importance of long-term projects in both collecting the data that is able to develop this type of deep learning models for individual identification. And in turn, it will be long-term projects themselves that will be mostly benefiting from these developments, as it will allow researchers to automatically collect individual data and potentially even collect data without the need to ever mark their study animals. I also want to take a few seconds just to acknowledge all the people that helped me over the year and to all of you that took the time to listen to my talk. Thank you very much.
Great, thank you. That was terrific. I think let's start with a question from Liz, and then I have one as well.
That was an amazing talk. I'm curious what you think the future of data collection is in animal behavior. Specifically, can these individual identification deep learning things be combined with, like, I'm assuming they can, with automated data collection where you wouldn't even have someone like count how many visits you have, just the computer does everything?
Yes, so I think that we can never replace the human factor. So I think that everything starts with a human observation. So we still have to observe our animals to understand how we are going to collect the data. But then when we get to the point where we are on that repetitive stage of collecting the same data over and over again, I think that can be completely replaced by a machine. And I think it should, because it will allow researchers to have more time to think about their questions, to analyze their data, to study the literature and so on, instead of spending endless hours analyzing, I don't know, nesting provisioning rates and stuff like that.
Thanks.
And so my question's pretty nuts and bolts. I'm curious if you can tell us, does the model tell you what features of the birds it's using to identify them? And if so, what features is it using? Not
directly. So
okay.
that you would have to either apply specific methods that have been developed to do that. But most these deep learning models are known for being a black box where you don't actually know what is looking at. It is possible to know, but it requires additional analysis. You cannot know them directly. So I don't know exactly what he's using to identify these birds.
Got it. And so I guess that might, like when you showed that picture of the conserved elements, the relatively conserved elements over time, by the time we get to like the deep elements, like to me, it doesn't even look like a human face anymore. So I guess there's not a question there really, but I'm curious just to hear your thoughts about how you think these training models line up with human intuition and human perceived differences versus, to the extent to which this is something completely different that we would otherwise miss.
I think they don't line up. I think they are really abstract to our understanding. So it's true in that example. It seems like it's not a human face anymore, but I think that's just really illustrative of what is going on.
Great. Well, I think we'll leave things there. Thanks so much for that talk, André.
Thank you.
Our second speaker today is Dr. David Fisher, a research fellow at the University of Aberdeen. David completed his master's degree at the University of Liverpool and his PhD at the University of Exeter before holding postdoctoral fellow positions at the University of Guelph and McMaster University, and now at the University of Aberdeen. And David has studied social behavior, senescence, quantitative genetics, and really a wide range of social questions in a range of systems from spiders to poison frogs, and in long-term studies of red squirrels and crickets. He's made contributions both to empirical and theoretical understanding of social behavioral evolution, including multi-layer spatial networks and multi-level selection, two topics that are joined by their theoretical complexity and their insistence that we expand our evolutionary thinking beyond those things that are most readily observable. And I think that wide range of curiosity and willingness to see beneath the top layers of questions and systems is a strong motif in David's work. And so I'm glad to welcome him here, and I'll turn things over to him now. You're currently muted, Dave.
I've watched so many people do that in presentations online. I'm thinking, like, oh, we've been doing this for 2 years, folks. Have you not got this yet? And then, of course, I do that immediately myself. All right, great. Yeah, thank you very much for the introduction. It's exceptionally kind of you. So I'm going to be talking about social networks and antisocial animals. So I'd like to emphasize that although I might be in the longer slot here, I'm not the PI on any of the work I'm going to talk about here. This was my kind of early career research. And so I'm going to kind of note out who the kind of senior scientists were in each case. So I'm really interested in animal social interactions. These are the kind of cooperative interactions that animals engage in their day-to-day lives, such as these penguins huddling together for warmth. But social interactions can also be competitive. So these very same organisms can then be competing with each other for access to resources in one environment while cooperating in another. So it means a really important part of organisms' environment is their social environment. So it's conspecific to both cooperate with and compete with. So we can represent this social environment as a network. So each individual penguin here will be represented as a node, and then these interactions or associations or relationships with other individuals will be represented as a line between them. And under the hood, this network can be thought of as a matrix of pairwise interactions. So this matrix just demonstrates that the interaction between A and B was present or a strength of 1, whereas the interaction between A and D was absent, so a strength of 0. So we can use social network analysis to analyze these matrices and analyze these networks, and they're a really useful tool for quantifying the social environment. So yeah, as I said, you have these nodes and they're linked by edges. There are other terms in network science like vertices and so on, but I won't go into those now. And you can, these matrix of relationships can be represented as binary, so presence or absence of the relationship. But you can also weight them. So these ones could have values to represent the strength of an association or relationship. And that means you've got a really, really flexible approach here for quantifying different kinds of social interactions. So for instance, we can quantify the number of times this mammal licks the windscreen or grooms one of its conspecifics and kind of use that to inform a weighted network of affiliative social interactions. We can also see how these birds here, how often they're seen in flocks together, and so create a network based on those relationship strengths, representing birds that frequently associate or frequently flock together compared to ones that never do or do only occasionally. So a lot of research on these kind of social networks has focused on these long-lived vertebrates because they're relatively easy to mark and observe, and so you can see them interacting with each other over time as they build up a network of social relationships. As you can see a dolphin example here, these animals have probably been living for quite a long time, maybe even decades, and they've been able to observing each of these uniquely identifiable individuals to be able to build up this kind of detailed list of associations. As an aside, I really want to meet the biologist who called dolphin fish. So typically, only long-term studies have these kind of data where you are able to observe the same individuals repeatedly to build up a complex network of social interactions. And most of the time, they tended to focus on cooperative or at least affiliative social interactions like grooming or flocking together or co-occurring in groups. But there's absolutely no reason why you can't use the same approach using social network analysis to look at competitive interactions as well. And competition is likely to be as important, if not more important, for many animals around the world. So I think it's a really useful opportunity to use social network analysis to quantify competitive interactions and then see what role in different ecological and evolutionary processes they play. So, for instance, you could have these two eagles there interacting over a prey carcass, and although they're solitary and they do not typically move or act in groups, you can still build up a matrix of interactions when these individuals compete with each other for access to resources, and you could use that to explore how some individuals have access to lots of resources and some individuals have access to few resources. Now, I wasn't working on anything quite as majestic as an eagle. I was working on something far more interesting, which was a cricket, and specifically these crickets. So this is Gryllus campestris, a European field cricket. This is a male here, and he uses these little wings. He rubs them together to sing and attract females. And this female here, she's a bit larger, and you can see her ovipositor at the back. So I was interested in using social network analysis to see how competitive interactions amongst these field crickets influence their lives and selection and so on. So this is part of the Wild Crickets project where I did my PhD. And this is a truly amazing study system where in a meadow in northern Spain, each cricket burrow is marked with a little white flag, which you can see here. So there's typically several hundred of these in a given season. And then over the most active burrows, these cameras are mounted to film the interactions the crickets are engaging with. And so you build up over 100 cameras, an enormous amount of footage of individually marked insects interacting. So this project is run by my two PhD supervisors, Tom DeGenza and Rolando Rodriguez Munoz, and without their dedication and hard work to get this project up and running, and then their guidance while I was working with them, none of this work would have been possible at all. So as I said, we have individually marked crickets, and you can see that here. So we've got male 1C is hanging out at a burrow with female 66, and this is burrow 121. So this little green stick here is the base of one of these flags here, just to give you an idea of scale. And so we can observe males and females hanging out at burrows and mating, and so look at how those social interactions are occurring. But more personally, for the topic of this talk, we can also look at competitive interactions. So here we have male CN fighting another cricket who we can't see the tag of. You can see their mandibles are flaring widely and they're dashing into each other with great zeal. And so we can directly observe these fighting interactions and so use them to make a network of that kind of interaction. But simultaneously, as I said, we're also observing males mating with females. And the footage is good enough to see when a spermatophore is passed from a male to the base of the female ovipositor here. So we can see when a male has successfully mated with a female. Now, females are polyandrous, and we know they store sperm. So that means we can infer which males have mated with the same female, and therefore who it might be in sperm competition with each other. So we have males competing in two different arenas. They are physically competing with each other by fighting, and they are also competing with each other in sperm competition by mating with the same females. And we can represent both these kinds of interactions as networks. So this is a social network of fighting interactions. And unlike a lot of social network plots, I've located the individuals based on the coordinates of the burrow they were closed in. So this male started out here, way out on the left side of the meadow, and then moved further in and interacted. And you can see some individuals don't get much fighting at all, or some are in the thick of it and engage in lots of different fighting interactions. And then simultaneously, we can look at the exact same males. The positions are exactly the same between those two plots. But this time, males are linked if they both successfully mated with the same female and are therefore in sperm competition. Now, then you can see that some of these links are present in both. So you can see this blue individual at the top here has got lots of links, and some of them are present in the fighting network as well, but not all of them. And so some individuals are well connected in one network and not the other. And connected with some individuals and not others in that same network. So I wanted to analyze these two different networks and compare them and see what they tells us about pre-copulatory competition, which is these fighting interactions, and how it's related to post-copulatory competition, which is this network of sperm competition. So what we first see is that males tend to fight those they are in sperm competition with. So I initially thought that by fighting another individual, so this interaction here, they'll be able to avoid being in sperm competition with them because one of you would win that fight and then the other one would leave the area and not mate with the same females. But in fact, the opposite is true. Once we account for overlap in space and time, there's a positive association between the two networks, which means that males are fighting the same individuals they're in sperm competition with. And I think this is probably because these kind of males are going to be quite similar in mass or in fighting ability, and so they can't separate each other by fighting, and neither can the females can't necessarily choose between them either. So they end up both fighting and being in sperm competition. Secondly, we see that males who fight more, so have more different rivals in the fighting network on the x-axis, tend to be in sperm competition with more different males. So this is the y-axis here, and the different colored dots are the 2 different years we did this analysis. So that means we don't have any kind of specialization where some crickets focus on being really good at fighting and less engaged in sperm competition, and vice versa. What we see instead is that the males who tend to fight lots of different other males are also in sperm competition with lots of different other males. And this could be to do with differences in activity level or vigor, or differences in lifespan that allow them to be heavily engaged in both types of interaction. And finally, when we look at the relationship between males and females, we see assortative mating by promiscuity. And so what that means is that males with many different female partners, those females tend to have many different male partners. And this is clearly different from the null expectation in 2006, and not positive, but not different from the null expectation in 2013. So males with many mating partners mate with females who would mate with many males. And what this does is this should reduce the variance in reproductive success in the population, because the males, and the males in particular for males, because males who appear to be doing well, mating with lots of different females, they are having to share fertilization success in each of those females with all her other mates, which means they're going to get a smaller share of each fertilizer, of each set of her eggs, compared to males that are only mating a few times, but they have a higher exclusivity with those females, and say they should fertilize a higher proportion of those eggs. So that work is in the journal Behavioral Ecology, and as I said, it was with Rolando Rodriguez-Munoz and Tom Tigenza, and throughout my PhD I was funded by NERC, so I'm very grateful for that opportunity. So this is interesting. So we've got these different kind of fighting and kind of competitive interactions going on, and they seem to kind of be related to kind of activity or vigor or something like that. But I was also interested in how these fighting interactions directly relate to subsequent mating interactions crickets might have. So any given male cricket is both fighting with other males and trying to engage in mating opportunities with females. So these lines of different colors here representing the different kinds of interactions. So we can represent these fighting interactions as a network, and you can see here the different colored nodes are males and females. So females in this species do fight each other for access to burrows. So we see males fighting males and females fighting females the vast majority of the time. And then we can also equivalently represent the mating interactions as a network. So here you can see the different color nodes are only joined to each other. So males only connect with females and not males to males and females to females. So then we can see how these two different networks dynamically influence each other. So does fighting on one period of time then lead to mating opportunities in the next period of time, or do in fact need to conserve energy and so on? To approach this problem, I use stochastic action-oriented models, which are a type of dynamic network analysis really useful for seeing how interactions influence traits and other types of interactions over time. So we ended up writing a paper about this because I thought this would be really useful for behavioral ecologists, and that was work with Amiel Alani, Matthew Silk, and Tom Tigenza. And Amiel in particular knew a lot more about these methods before we started, so I'm really grateful for his help, and Matt Silk as well, who I've worked on a lot of different networking stuff with. So we can then use this approach to answer several questions. So we know that both social network connections in any normal network and mating success in a typical population are highly skewed. So on the x-axis we here have the number of different mating partners and then the frequency, and you'll see this in almost any wild population. The vast majority of individuals have none or very few mating partners and low mating success, and then some individuals are incredibly successful. So if both networks and reproductive success in general tend to have this property, then can we model mating success as a network and therefore try and explain how we can get this kind of great skew in fitness in populations, which of course underpins evolutionary change? And simultaneously, we can also look at how fighting and mating are dynamically related. So does mating lead to fighting? Does fighting lead to mating? And so on. So what we found was that the fighting network was influenced by proximity. So individuals near each other are more likely to fight than individuals far apart, if it makes perfect sense. It also different between the sexes, and actually given their opportunities, females are actually more likely to fight than males. It's possibly because they're fighting over access to burrows, which are incredibly important for surviving long periods of time. We also found a mass influence of fighting networks, so heavier individuals are more likely to engage in fighting than lighter individuals, possibly playing to their strengths there. And finally, mutual connections were important in the fighting network. So individuals that shared a mutual connection were more likely to fight each other than individuals that didn't share a mutual connection. And given we've already controlled the space, this is a bit puzzling, but I think it could be something to do with sets of similar males who can't quite work out a dominance hierarchy between them, constantly fighting each other. And interestingly, the fighting network did not influence the mating network or vice versa in this system. So engaging in fights did not necessarily lead to mating opportunities in the next time period and vice versa. What we did find was that only positive assortment by mating success was required to model the mating network. And this is a bit similar to what we saw earlier with the positive assortment by promiscuity, in that males who are highly successful at one time point are going to be even more successful at the next time point, and so on, and the same is true for females. And so this kind of positive feedback loop then helps create this great skew in mating connections, and therefore probably in fitness in the population. And so that work is published in Animal Behavior. And so I think stochastic active-oriented models are a really, really great and useful tool for analyzing interesting questions in behavioral ecology. So check out the previous paper and this one if you want to know more about the different ways you can use them. So we've got these networks of fighting, networks of sperm competition, networks of mating interactions, and it's appear interesting. But what would be more interesting is if they were stable over evolutionary time. And what I mean by that is the structure of these networks was similar from one generation to the next. Because if that's true, then these networks are going to apply consistent selection to our population and therefore cause it to evolve in a particular direction. Because in contrast, if the social network changes fundamentally in structure from one generation to the next, then you're going to see different kind of types of selection represented by different color arrows here acting on our population. And so you're not going to see evolution in a consistent direction due to these social networks. Now, here, the crickets have a great strength over most long-term studies in that each year we get a new generation of crickets. So the study has been running since about 2006. Every single year, that's a generation. And so we have, and there's no overlap between individuals. The adult will die before the young hatch. And so we have multiple independent networks we can use to determine whether networks are consistent and stable across generations. You can't do in most long-term studies because you have the same individuals still alive across years, and they're going to be influenced by the network structure. So to do this, to see how networks were similar or different over time, I fitted exponential random graph models to each year. So exponential random graph models are another technique Matt and I think are going to be really, really useful for understanding animal social structure, because you can model so many different processes at once and really get a good understanding of what drives social network structure. And so what we could do is we could fit the same exponential random graph model to each year and then use the parameters from that model to simulate the network structure in other years based on the demographic parameters in that year. And then we could see how similar the network based on the rules of one year was compared to the structure of the network in that other year. And then we could see if the number of years apart predicted this network similarity, because if it does, that suggests the networks are gradually changing over time because you have more years close together and more similar, and years far apart more different. An alternative hypothesis was that population size determines the kind of interactions that are happening in the population, and therefore similarity in population size will then predict network similarity. So the first of those are these two plots are for two different measures of network structure. One, path length, is kind of a global measure of how well connected the network is, and then clustering coefficient is a more local measure of how tightly individuals are clustered with their mutual connections. But in both cases, the number of years apart two networks were, which is shown on the x-axis here, did not predict how similar the two networks were for both path length and clustering coefficient. So the answer to this question here is no. Number of years apart does not predict network similarity. And then what instead we see is that the difference in population size is positively associated with how different the networks were. So populations that are very, very different in size have much more different networks, and populations that are very similar in size have a much more similar networks. I think you can kind of see that here. So we've plot of five of the years I analysed here. You can see that the kind of lower population density years, kind of these two, you have these more kind of spread out networks without major hubs. Whereas in the more dense population years, you have these real central areas of high intensity interaction, and then these kind of like isolated individuals out on the edge there. So that work is in BMC evolutionary biology, if you want to see how exponential random graph models can be used to kind of answer interesting questions about network comparison. So it's cool that these networks of competitive and mating interactions are playing a role in the lives of these crickets. And I think that because they're reasonably stable over time, that they should be able to impose meaningful selection. So then kind of a bigger question, this is what I finished my PhD thinking, was, okay, how do these networks of interactions actually influence evolutionary change? How can we quantify how these networks of interactions influence how phenotypes change from one generation to the next? And so I ended up answering this question in squirrels. Squirrels, you might be thinking, that seems a bit strange, especially for me, because most of the work I've done has been on invertebrates, such as insects and spiders. But I got the opportunity to work on this rather cute little organism here, the North American red squirrel, Tamiascurus hudsonicus, and this is part of the Kluane red squirrel project. This is a long-term study that's been running since 1987 in the northwest of Canada. And you can individually mark squirrels with these ear tags here, and so you can recognize individuals when you trap them. This is a postdoc I did with Andrew McAdam, and I'm incredibly grateful for Andrew for giving me the opportunity to work in his lab group and to work on this long-term data set. It was a really, really amazing opportunity, especially because the data collection is really quite intensive, and so they can monitor when female squirrels give birth. You can see here one carrying a newborn pup in her nest there. And that means you can collect really, really detailed life history data. And thanks to these observations, we also know where squirrels live in the forest. So each squirrel, males and females, guard their own territories in the forest where they cache resources to survive the winter. Excuse me. And this map here shows the locations of several different territories, each one being marked out in a different color. This allows us to see who's living next to who, and therefore, who, even in the solitary organism, who could be engaging in competitive interactions. Now, thanks to these detailed observations of a mother and her pup, we've had a maternal pedigree going back to the very start of the project. And thanks to genetic analysis in later years, we've been able to identify fathers as well. So a really, really deep pedigree, and that allows us to answer various interesting questions with a much more kind of evolutionary angle about the genetic component underlying interactions and so on. And so I'm going to take you through a little schematic to introduce one of the topics here. So we can think about any particular squirrel's traits, so its body mass or its behavior, is being influenced by its own genes and the environment it experiences. And the effect of its own genes on its phenotype, we can think of as a direct genetic effect. That's this here. But when other individuals are in the environment and they're playing an important role, these other individuals are also influenced by their own genes and their own environment. Now, if this second individual influences our focal individual through social interactions in some way, and this depends on the phenotype of our current individual, that means that the genes of our partner individual are able to influence the phenotype of our first individual, mediated by social interactions. And these are called indirect genetic effects. We've got the direct genetic effect with the genes of the organism influencing itself. The indirect effect is the genes of another organism via social interactions influencing our focal individual. And these indirect genetic effects can dramatically alter the direction and the tempo of evolution because they provide a whole new source of genetic variation, which can greatly increase how quickly traits can evolve. But they can also constrain the evolution of traits. Because if there is a negative correlation between direct and indirect genetic effects, and that's shown here, let's say an individual with genes that tend to give it a high trait value, so these plus symbols here, maybe through some kind of competitive interactions, it causes individuals that it interacts with to have lower trait values, these minus signs here. Now, if in the next generation, this individual does particularly well and so has lots and lots of offspring, they're all going to be like this, with the genes that have high trait values, but also the genes to cause other individuals to have low trait values. And so, although you might expect the next generation to have an overall increase in the trait mean, because all the individuals have the genes that have a high trait value, you've also got a population full of individuals who are negatively influencing the traits of their neighbors. And so, overall, the trait mean may not change much, it may not change at all, or it could even go back, reduce. This is how this negative correlation between direct and indirect effects means that IGEs can cause evolution to go backwards, which I think is really, really cool. Now, these kind of indirect effects are really well appreciated in livestock, interacting in dense groups in captivity, because they can tremendously influence yields. So chickens can peck each other to death, and pigs can interact and kind of reduce each other's growth rate and so on. And so people breeding livestock are really well aware of these kind of things. But they're less well appreciated among networks of wild animals. And that's partly because the methods to kind of quantify how a whole network of organisms are influencing each other are much more difficult to apply than how two individuals are interacting and influencing each other at a dyadic level. So I wanted to rectify this by adopting an approach from forestry genetics to think about how a squirrel's nearest neighbors, so which is a squirrel's 6 nearest neighbors, influencing its phenotype. So we use 25 years of data based on this long-term study, and fitted an animal model in the software ASREML. Now, unfortunately, as far as I'm aware, it's only the proprietary version of ASREML, which you have to pay for, that allows you to weight interactions by something like the distance or the strength of an association between two individuals. So I don't think the versions you can get on R or other types of analysis where can actually fit these models right now, which is a shame, but hopefully that will change over time. But we can use this approach from forestry genetics to account for the distance between neighbors, and so we can weight the interaction between these two more strongly than the interaction between these two because of the distances involved. Now, because we were looking at competition, traits such as the date a female squirrel gives birth to a handful of pups. Now, this is called parturition date. Now, this is a resource-dependent trait, and females who have more resources give birth early, and females who have fewer resources give birth late. And so we can make the prediction that if we looked at the strength of these interactions at high densities, you would have a stronger effect from the neighbors on the focal individual, and at low densities, you'd have a weak or no effect, because individuals are not going to be competing for resources as intensely at those low densities. And then we also, we expect stronger social effects at high densities compared to the social effects at low densities. And secondly, we also expected a negative direct-indirect covariance, because we think that if a squirrel has the genes to gather lots of resources and therefore reproduce early in the year, it will then be taking resources away from its neighbors and therefore negatively suppressing their parturition date. So negative covariance there. So I'm going to present the results from those models in a table form. So what we see first, the direct genetic variance, both high and low densities, it's there and it's significantly different from 0. And once you remove the among year variation, and leave behind the variation we think is pertinent to selection, you get a heritability of about 15%, which is in line with previous estimates. Then if we first look at how individuals are influencing each other at the phenotypic level, we see that in line with our predictions, there's a small amount of variation among individuals, how they influence their neighbors at low densities, and a larger and clearly from 0 amount of variation at high densities. So at high densities, some squirrels cause their neighbors to give birth early, and some squirrels cause the neighbors to give birth later. When we then split this phenotypic effect into its genetic and non-genetic components, we see a broadly similar pattern in that low densities, essentially no discernible direct genetic variance, and at high densities, there is probably some genetic variation in how individuals influence each other's parturition dates. I will note, though, that these estimates here with the stars are more than 2 standard errors away from 0. This one was between 1 and 2 standard errors away from 0. So it's not a really, really clear result. And this is despite a really, really large data set. So these indirect genetic effects are pretty difficult to estimate, especially in wild animals living on their own. With regards to our other prediction of the covariance, in line with our prediction, more or less, we see that at low densities, there's a positive covariance, and at high densities, there's a negative covariance. But neither of these were actually different from 0. So that's not particularly good support for that prediction. Now, what this indirect genetic variance does mean is that while at low density, there's approximately 5% of the overall phenotypic variation is to do with genes, at high densities, it's nearer 15% because of these effect neighbors are having on each other. So there's a greater evolutionary potential in the population at high densities than there is at low densities, due to the fact that individuals their genes influence the date their neighbors give birth. So that's a technical summary of what I've just said there, and you can read about that in this Journal of Evolution Biology article. I want to thank Stan Booten in particular, who's the main person who founded the Kluane Red Squirrel project been running since 1987, so without his hard work over these decades, none of this would have been possible. And then the other people involved in the long-term running of this project, Ben Dancer and Jeffrey Lane, their input on this paper was vital. Alistair Wilson, the second author here, he was instrumental in introducing me to some of these approaches to understanding how indirect genetic effects can work in populations of solitary organisms, and he gave some really insightful thoughts on this paper. And then David Coltman and Jamie Gorell helped out with the genetic analysis of the pedigree and so on. And of course, Andrew is my supervisor and kind of main person I was working with throughout. And this project was funded mostly by NSF and NSERC throughout. So this work shows that although we typically imagine and kind of use this model of the genotype influencing the phenotype, influencing fitness to help us understand how evolution works. But this work demonstrates that when other organisms are an important part of the environment, then we can disrupt this kind of simple pattern, because the genotype of one individual can influence the phenotype of another. But that's not the only pathway the only way we can kind of disrupt this simple pathway. So we'd also get a situation where the phenotype of one individual can influence the fitness of our focal individual. And this is called social or multi-level selection. And these two approaches are mathematically equivalent, and so you can kind of move between the two of them, which is really useful. And so again, I wanted to see whether a focal squirrel's fitness is being influenced by the phenotype of its near neighbors. In particular, we wanted to test the idea that a squirrel's interaction with its nearest neighbors should be much more intense, so a darker blue circle here, and then get progressively lighter as you consider wider and wider spatial scales. So what we see, so the trait we were looking at here was actually the trait of a juvenile squirrel, and its fitness measure was probability of recruiting. So did the squirrel survive over winter or not? And so the first kind of scale we looked at was the birth date of this litter relative to the average birth date in the neighborhood. And what we can see is that clearly for females and less clearly for males, but the interaction was not actually significant, so these two are not necessarily different from each other according to the model, was that individuals who are born earlier than the average birth date in their neighborhood, which is so on this end of the graph, they have a higher probability of recruiting. So sorry, the social neighborhood is all other litters within 130 meters. So what we think is going on here is that squirrels who are born earlier than their near neighbors are then able to get out and find a vacant territory and take it over before winter comes, and those without territories typically tend to die. And this effect gets stronger at high density. So when densities are higher, it's even more important to be born earlier than your other litters near you to be able to find the territory. When we consider then the average birth date of a social neighborhood compared to the birth date in a wider study area, we see an interesting interaction with sex. So for females, again, it's better to be in a neighborhood that's born early than your rivals at a wider geographic area. And so if you're born earlier than them, you have a higher chance of survival. But for males, in fact, it is opposite. So males who are born later than their wider rivals tend to have a high chance of recruiting. And so we think this is possibly due to kind of interesting patterns in how mother squirrels bequeath their territories to their offspring. So they occasionally do something called bequeathal, where a female squirrel will leave her territory to one of her offspring, and that tends to be a daughter, and that may favour them in that way. And then males may then benefit more from dispersing, and possibly if they disperse late, they have a high chance of survival. Now, when we start looking at even wider spatial scale, we start to see an interesting pattern. So if we compare the average birth date in the study area relative to the average birth date in a given year, we start seeing a positive relationship. So squirrels whose whole study area is born later in the year tend to be more likely to survive, which is opposite to what's going on here. And then we see an even stronger relationship with the among year comparison. So in years where everyone is born later, overall survival is higher. And in years where everyone is born early, survival tends to be lower. And I think this is to do with it being physiologically taxing to breed when colder. So that's what's going on here. So basically, squirrels who everyone tends to be born early they are in much colder environments, and therefore survival in general will be lower. But what that means is that it's still better to be born earlier than your close neighbors, especially at high densities. And that means there's a conflict in selection from the social and the physical environments. So the physical environment saying, hey, it's very, very cold, be born later in the year in a more benign environment. And then selection from the social environment is saying, look, be born early because we need to get out there before our rivals and claim a territory. And that work is published in Evolution. And again, along with the people I previously thanked, I also thank Murray Humphreys, who were working on the Squirrel project at the time of that article. And so I think it's really, really fascinating that these social selection pressures can be driving a trait in one direction, and other kinds of selection pressures could be driving a trait in a different direction. And you might expect that thing to leave the phenotype of our squirrel somewhere in the middle, although testing that idea hasn't been done yet, as far as I'm aware. And so these two pieces of work demonstrate how you can look at these competitive interactions among a kind of network of solitary individuals who don't live in groups, but still you can determine how their social interactions might be influencing evolutionary change. We can see that from that last bit, example, selection from a social environment may be moving in the opposite direction from other sources, and that the evolution of trait may be constrained by competitors, especially when resources are hard to come by. And from the previous work on the crickets, I showed you that different types of interactions can be compared and related using social network analysis. And that then allows us to quantify the social structure of asocial or antisocial species, because they're still engaging in important social interactions like competition for mates, competition for resources, and so on. I was especially pertinent for this seminar series that none of this would have been possible without a long-term study. In each case, you need to be studying marked individuals pretty much all of their adult life. And so you can quantify where they live, who they interact with, and what the consequences are for that. And then you can measure the fitness consequences and the things across generations. So really, long-term studies have a particular benefit for understanding how social interactions influence evolutionary change, which is kind of my main focus. But of course, simultaneously, none of this in terms of my career really would have been possible without long-term studies. And this is a picture of me before starting my PhD, trying to attempt to be with a flower. I was a really miserable attempt to be a biologist. And here's me after my PhD cutting a cake as a great success, because I was lucky enough to work in these long-term studies where you get really excellent training in all manners of kind of experimental design, data analysis, and so on. And this continued on to my postdoc, where I needed a book to work out what squirrel was, and then here I am actually studying them in the forest. And then, of course, it's greatly this experience of working on these different long-term studies has then influenced where I am now, doing field work on spiders in Ecuador, or even doing lab work on different organisms. And I really wouldn't be where I am now without long-term studies, the people who set them up and then supervise students and postdocs through them. So I really am grateful on a personal level for being involved in such things. So I'll leave you there with a summary of what I've talked about, and I will gladly take any questions.
Great, thank you very much, David. I have a couple of questions about the cricket system. So do you have paternity data? And so do you, are you able to assess to the extent to which sperm composition actually affects paternity and whether females are choosing somehow in a cryptic way?
So they do have paternity data for some years, yeah. So they can look at that. There was some early work in a science paper in 2010, which I think tried to get at this question. I mean, certainly we may know that females who mate many times then tend to have more offspring, and males who mate many times also tend to have more offspring. So there is still definitely some kind of very simple relationship there, but there definitely is the capacity for females in this species to engage in cryptic female choice based on the fact that they mate with several different males, but I don't think it's quite quantified exactly what role that plays.
I think Liz also has a question about crickets before we move to the Q&As about squirrels.
Sure.
Oh no, my question is about squirrels.
Go ahead.
I'll go ahead anyway. The crickets are really cool, but I'm interested in the indirect genetic effects. So you said that they were small, and you suggested that this, could be because it's really hard to measure these things in wild populations. And so I'm curious your thoughts on how we can better measure them in the wild, or if they really are so small in the wild. And then
Yeah.
following up on that, even if they are small, do you think that they're still important? Because yeah, I'm with you that I think, anyway. Yeah.
So in regards to are they small in the wild in general, I'm currently helping out with a meta-analysis with some folks. We're looking at indirect genetic effects. We're looking at both wild and captive studies. So we should be able to answer that question, like how prevalent are they typically in the wild? One of the great things about the general field of quantitative genetics is we have all these really useful equations we can plug our parameters into. So even if they are small, we can say, okay, they're going to influence phenotypic change this much. And then I think it's up to you whether you care about that amount of change or not. Obviously, a small estimate will end up being a small difference in considering IGs versus not considering them. But it could still, reduce or increase the speed of change depending on the correlations and so on. So in some situations it can matter. And because of what we could do differently, I don't know really. So I came across two papers on the great teeth of White and Wood where they quantified indirect genetic effects of the male on the female, and there was two papers that have done this. And they greatly increased the size of the data set from one iteration to the next. And the estimate was very similar, and the standard errors were even very similar. So it wasn't like the accuracy didn't increase that much. So I was just like, oh, what's going on? Like you think, oh, more data is always the answer. In that situation, maybe it wasn't. So I think we need to change and look at more different organisms. I think if we look at organisms which form small groups with tight associations, I think you're much more likely to find a signal for indirect genetic effects there, where it really, really matters who you're interacting with, versus kind of the squirrels and the great chiefs, where they become these big, loose aggregations or networks where any given relationship is quite weak. But that's of course that's stacking the deck in your favor, right? If you want to find strong indirect effects, you go look in the system where you think particular social relationships are really strong, which doesn't necessarily mean that they're going to be generally important for everyone else's system. But I mean, I think that'd be useful. There's still not enough. They're a bit niche. And so I think more different organisms, if people have the data to quantify them, and it'd be really, really useful. Especially because I'm doing a meta-analysis. So more estimates are always handy.
Yeah, thanks.
Great. And then I think let's close with this question from Alicia Brody, who asks, are there any strategies that red squirrel mothers use to mitigate the conflict between the effective parturition date on sons and daughters? And does this vary by high and low density years?
In terms of mechanisms, well, like we know the females engage in bequeathal, and so they will sometimes leave their territory behind, and more often than not, it's a daughter that gets it, although not always. And we don't know why this is. It could be because the females are a safer bet, and that males typically maybe have more variance and we've got success, and so it makes more sense to put your eggs in the female basket rather than the males. Because males can maybe disperse further and may get very, very lucky and have a really, really kind of high fitness, but then may also always die. But beyond that, no, I don't know. Sorry, people have been working. People in Ben Dancer's groups with Sarah Westrick have been doing more on the paternal behaviour of the red squirrels, and so you can look into that if you're curious about what they're getting up to. But I'm not sure about any work in particular related to the kind of these different effects of neighbours on the two sexes.
Well, I think we'll leave things there. Thanks both to André and David for giving two really fascinating talks today. And to everyone else out there, thanks for coming. We'll see you same time, same place next week.
Thanks for having me.
Great. Thanks. Great to see you.